Friends, we are discussing first step of genetic engineering, that is isolation of desired gene, right? In this isolation of desired gene, we are discussing about restriction enzymes. What's a restriction enzymes? Is it okay, right? Now listen here. The credit of discovery of restriction enzymes. The credit of discovery of restriction enzymes, the credit of discovery of restriction enzymes goes to number one, Arbor. Scientist names I am writing, right? Arbor, Smith and Nathans, right? Therefore, the credit of discovery of what's a restriction enzymes goes to Arbor, Smith and Nathans. Sir, these three scientists they are called as a what? Trio of restriction enzymes. These three scientists they were called as what? Trio of restriction enzymes. Who are called as a trio? Trio means three, right? Who are called as a trio of restriction enzymes? Arbor, Smith and Nathans. Is it okay? Right. Now, sir, what the term restriction means? In restriction endonucleases or restriction enzymes, the term what's a restriction? The term restriction in restriction enzymes or restriction endonucleases. Restriction endonucleases refers to preventing what? Preventing a growth of bacteriophage. Preventing growth of what, sir? Bacteriophage. Preventing growth of bacteriophage in host bacteria. Is it okay? Sir, restriction enzymes are in restriction endonucleases. What this term restriction refers to? This term restriction refers to preventing growth of bacteriophage, preventing the growth of bacteriophage in host bacteria, preventing growth of bacteriophage in a host bacteria. Is this statement okay? Right? Now listen this statement. Sir, we are discussing which enzymes? Restriction enzymes, right? Restriction enzymes cuts the DNA by two ways. Restriction endonucleases cuts what? DNA by how many ways, sir? Two ways. Cuts the DNA by two ways. What are the two ways, sir? Number one, even cut. Second one is a staggered cut. What are the two types of cuts? Even cut and staggered cut, right? Go for the first one. What it is? Even cut. Go for even cut. Now see here, I am taking a double-stranded DNA molecule. Double-stranded DNA molecule means with how many strands, sir? With two strands, right? For example, a restriction endonuclease has made a cut in the first strand at this point. The restriction endonuclease is also cutting another strand of DNA at the same point. Therefore, cuts both the strands of DNA at the same point. What is that cut called as? Even cut. What is that called as? Even cut. Now listen, sir, if restriction endonuclease enzyme cuts at this point and cuts this strand at this point, then what happens, sir? We will get fragments, yes or no? What we are going to get, sir? We are going to get fragments. We are going to get the fragments. See this end, right? This end is called as blunt end or flush end. Is it okay? Therefore, when even cut is made, right, ends will be either blunt ends or flush ends. Blunt ends or flush ends. Is this statements okay? Right? Therefore, which cuts are? This is even cut. Restriction endonucleases. Restriction endonucleases cuts both the strands of a double-stranded DNA at same point. Therefore, cutting what, sir? Both strands at the same point generates which type of ends? Blunt ends or flush ends. Therefore, even cut, right? Even cut produces which ends, sir? Blunt ends. Blunt ends. Sir, blunt ends also called as flush ends. Is it okay? Right? Therefore, even cut produces which ends, sir? Blunt ends or flush ends. Blunt ends or flush ends. Is it right? Right. Now, listen here. Sir, we are discussing even cut, right? Sir, which restriction endonucleases makes even cut, sir? Restriction endonucleases which makes even cut. Restriction endonucleases which makes what, sir? Which makes even cut. Which makes what, sir? Even cut. Examples I am writing, right? Number 1, SMA1, right? Next one, PVU2. Next, HAE, right? 3. Next, ECOR5. ECOR5. These are what, sir? Restriction endonucleases. These are what? Restriction endonucleases which makes even cut. These are restriction endonucleases which makes which cut? Even cut. Is this statement okay? Right. Now listen here. Sir, take ECOR5. For ECOR5, the recognition sequence, the recognition site is GATATC. What is a recognition site or restriction site for ECOR5, sir? GATATC. From which prime to which prime? 5 prime to 3 prime. From 5 prime to 3 prime. Is this statement okay? Right. Sir, G pairs with what, sir? G pairs with C, right? Sir, A pairs with what, sir? A pairs with T. T pairs with A. A pairs with T. T pairs with A. Right. Sir, C pairs with what, sir? C pairs with G. Is it okay? Right. Now, sir, what is this one, sir? This is restriction site or recognition site for ECO R5. This is restriction site or recognition site for which enzymes are ECO R5. ECO R5. Sir, ECO R5 makes which cut? It makes even cut. It means cuts both the strands at the same point. Sir, it is going to cut first strand at this point, second strand at this point. It means cutting both the strands at the same point. Cutting both the strands at the same point, that is in between T and A. In between T and A. Yes or no? Therefore, ECO R5, right, it is cutting the restriction site or recognition site at the center. At what? Center. At the center. Is this statement okay? Clear? Right. Now listen here. We are discussing which cuts are even cut. Which cuts are even cut. Restriction endonucleases cuts both the strands of a double-stranded DNA at the same point. Produces which ends are? Produces blunt ends or flush ends. Blunt ends or flush ends. Restriction endonucleases which makes even cut. Examples: SMA1, PVU2, HAE3, right, ECO R5. These are the restriction endonucleases which makes which cuts are even cut. Is it okay? Right. Sir, during even cut. During which cuts are even cut? Right. Restriction endonuclease cuts right where sir? At the center. Therefore, restriction endonucleases right cuts where sir? At the center. Cuts at the center of what? Recognition site. Center of which site sir? Recognition site. Therefore, in the center of recognition site, it is going to make a cut. Which type of cut? Even cut. Is this statement clear? Right. Sir, 5' to 3'. GATATC. This is what sir? Restriction site or recognition site for ECO R5. GATATC, that is from 5' to 3'. This is the restriction site or recognition site for ECO R5. ECO R5. Is this statement okay? ECO R5, it is cutting both the strands at the same point. That is where? Center of restriction site or recognition site. That is in between T and A. In between T and A. Is this statement okay? Clear? Right? Staggered cut. What it is? Staggered cut. Restriction endonuclease. Restriction endonucleases cuts two strands. Cuts two strands of a double-stranded DNA at two different points. Two different points, but between same nucleotides. But between same nucleotides. See here, sir, restriction endonucleases, right? Sir, this restriction endonucleases, they are cutting one strand or both the strands? They are cutting both the strands. Right. Sir, do they cut both the strands at the same point? No, at two different points they make the cut. But they are cutting between what sir? Same nucleotides. Cutting between the same nucleotides. Is this statement okay? With example, you can understand it more clearly. Listen, which cut this is? Staggered cut. For example, right, take a ECO R1. Sir, ECO R1, what it is? ECO R1. ECO R1, right, is a restriction endonuclease. It makes a staggered cut. ECO R1 is a restriction endonuclease which makes what sir? Staggered cut. Is it okay? Right. Now, sir, for ECO R1, what is a recognition site? For ECO R1, what sir? Recognition site or restriction site. For ECO R1, the recognition site or restriction site is a GA A T T C. Is it okay? What is the recognition site or restriction site for eco R1, sir? GAATTC. That too, it has to read from which prime to which prime? 5 prime to 3 prime. It means the sequence which reads from GAATTC, the sequence which reads GAATTC from 5 prime to 3 prime, only such sequences are recognized by eco R1. Therefore, eco R1 recognizes which sequence? The sequence which reads GAATTC from which prime to which prime? 5 prime to 3 prime. Sir, sequence is GAATTC, sir, but reading from 3 prime to 5 prime, sir, not recognized by eco R1. Therefore, sequence should be GAATTC and it should read from where to where? 5 prime to 3 prime. Then only it is recognized by what, sir? Eco R1. Is this statement okay? Therefore, what is the recognition site or restriction site of eco R1? GAATTC, reading from 5 prime to 3 prime. Now, you write its complementary sequence. What it becomes? C T T A G. Is it okay? Right? Now this is 3 prime to what's 5 prime. 3 prime to 5 prime. Is it okay? Right? Now listen this statement. Sir, restriction endonucleases, we are discussing which cut? Staggered cut. Sir, restriction endonucleases, they are cutting both the strands at same point or different points? Different points, but between the same two nucleotides, yes or no? Sir, eco R1 cuts it is cutting first strand at this point, cutting the second strand at this point. See, is eco R1 cutting both the strands at the same point? No. Cutting first strand at this point, cutting the second strand at this point, therefore cuts 2 strands at 2 different points, but between the same nucleotides. Sir, here cut is made between G and A. Here also cut is made between what and what, sir? G and A. Therefore, cuts 2 strands at 2 different points, but between the same nucleotides. Between the same nucleotides. Now you tell me, sir, what is the recognition site for eco R1? GAATTC, reading from 5 prime to 3 prime. Eco R1 makes which cut? Staggered cut. Eco R1 makes which cut? Staggered cut. It means cuts 2 strands at 2 different points, but between the same nucleotides. Between the same nucleotides. Is this statement okay? Clear? Right? Now listen. See here. Sir, staggered cut, right? Sir, here cut is made in the center of the restriction site or not? No. Here cut is not made in the center of the recognition site or restriction site. Here cut is made slightly away from the center of the restriction site or recognition site. Is it okay? We are discussing which cuts are staggered cut. Therefore, cut made where? Slightly away from the center. From the center. Therefore, cut made slightly away from the center within restriction site. Sir, cut is made in the restriction site only. Within the restriction site only cut is made. But is this cut made in the center? No, it is made slightly away from the center. Is this statement okay? Clear? Right. Therefore, sir, we are discussing which cut, sir? Staggered cut. Now, for example, if you take this case, right? Sir, cut is made here. Sir, cut is made here, right? Sir, if cut is made here, then what we are going to get once you see? G, C, T, T, A, right? This is one fragment, this is one fragment. Then what will be the another fragment, sir? A, T, T, C, G. This will be another fragment. You can check. Sir, this is one fragment. This is one fragment. Yes or no? This is one fragment. Whereas this is another fragment. You see, G, C, T, T, A, one fragment. Right? A, T, T, C, G. This is another fragment. Now see this fragment. In this fragment, this end came out. Paired or unpaired? Unpaired. So we are calling this end as protruding ends, sticky ends, cohesive ends. Therefore, staggered cut generates which ends? Staggered cut generates protruding ends or sticky ends or which ends are cohesive ends. Therefore, this is protruding end or sticky end or cohesive end. Protruding end, sticky end or cohesive ends. Is this statement okay? Right? Therefore, which cut we are discussing? Staggered cut generates which ends, sir? Protruding ends. Protruding ends or sticky ends or cohesive ends. Is it okay? Right? Therefore, sir, even cut, staggered cut. Even cut cuts two strands at the same point. Cuts two strands at two different points but between the same nucleotides. Even cut. Cut is made in the center of the restriction site. Cut is made slightly away from the center of the restriction site. Is it okay? Even cut generates blunt ends or flush ends, whereas staggered cut generates protruding ends or sticky ends or what are cohesive ends. Most of the restriction endonucleases makes a staggered cut. Therefore, mostly which cut is made? Staggered cut is made. Is this statement okay? Therefore, what, sir? Most of the what? Restriction endonucleases. Most of the restriction endonucleases makes which cut, sir? Most of the restriction endonucleases makes which cut? Staggered cut. Most of the restriction endonucleases makes a staggered cut. Is this statement clear? Is it okay? Right? See, here some important restriction endonuclease enzymes names are written. Is it okay? Sir, more than 900 restriction endonuclease enzymes are isolated till now, sir. Why we are discussing only these 8 types of restriction endonuclease enzymes here, sir? Because in the next coming topics, we are going to discuss one vector by name PBR 322. What is that vector name? PBR 322. This PBR 322 vector, on this PBR 322 vector, these 8 types of restriction endonucleases can act. That's why we are discussing these 8 types of restriction endonuclease enzymes here. Is it okay? In the next coming topics, we are going to discuss about PBR 322 vector. On this PBR 322 vector, these 8 types of restriction endonuclease enzymes can act. It means the recognition sites for these 8 types of restriction endonuclease enzymes are present on PBR 322. Yes or no? Sir, ECO R1 acts on PBR 322. It means on PBR 322, the recognition site for ECO R1 is present. Sir, BAMH1 can act on PBR 322. but BAMH1, right, it cuts the DNA at a specific site. That specific site should be on PBR322. Therefore, recognition site of BAMH1 is on PBR322. Is it okay? Sir, I am saying that these 8 types of restriction endonucleases can act on PBR322. It means for these 8 types of restriction endonucleases, right, restriction sites or recognition sites are present on PBR322. Is this statement okay? Right. Now, listen here. Sir, EcoR1, it is isolated from Escherichia coli bacteria. Isolated from Escherichia coli bacteria. BAMH1 is isolated from Bacillus amyloliquefaciens. Amyloliquefaciens. Is it okay? HIND3, Haemophilus influenza. HIND3 is isolated from which bacteria? Haemophilus influenza bacteria. PVU1 is isolated from Proteus vulgaris. Proteus vulgaris. PVU2 is isolated from Proteus vulgaris bacteria. Right. PST1 is isolated from Providencia, right, Sturti. Providencia Sturti. Next, SAL1 is isolated from Streptomyces albus. ALBUS. Streptomyces albus. CLA1 is isolated from Caryophyllum lactum. Caryophyllum lactum. Therefore, all these are restriction endonucleases. Then what are these, sir? This is the source from where this enzyme is isolated from. Therefore, E. coli is isolated from EcoR1 is isolated from E. coli bacteria. Is this statement okay? Clear? Right. Sir, EcoR1. What is a recognition site? GAATTC. Is it okay? Right. Sir, for EcoR1, what is the recognition site? GAATTC. Reading from 5' to 3'. EcoR1 cuts between what and what? G and A. Therefore, this arrow indicates cut. What this arrow indicates, sir? Cut. Is it okay? Right. BAMH1. BAMH1. For this restriction endonuclease, what is the recognition site, sir? G, A, T, C, C. Reading from 5' to 3'. BAMH1 cuts between what and what? G and G. Cuts between G and G. Now, HIND3 is a restriction endonuclease. For this enzyme, what is the recognition site, sir? AGCTT. Reading from 5' to 3'. This HIND3 cuts between what and what? A and A. Cuts between A and A. Now, PVU1. PVU1. What is its recognition site? C, G, A, T, C, G. Reading from 5' to 3'. Cuts between what, sir? T and C. Cuts between what and what, sir? T and C. Cuts between T and C. Go for the next one. PVU2. PVU2. Sir, PVU2 recognition site. What, sir? C, A, G, C, T, G. Reading from 5' to 3'. Cuts between what, sir? Cuts between G and C. Cuts between what, sir? G and C. Therefore, what this arrows indicates? Cut. Okay, right. Now go for PST1. What, sir? PST1. Sir, this PST1, right, for this enzyme, the recognition site is C, T, G, C, A, G. Reading from 5' to 3'. Cut is made between A and G. Cut is made between A and G. Cut is made between A and G. Go for the next one. SAL1. Sir, SAL1. For this enzyme, the recognition site is G, T, C, G, A, C. Cut is made between G and T. Cut is made between what, sir? G and T. Okay. Now go for the last one. CLA1. CLA1. Sir, for this enzyme, the recognition site is what? A, T, C, G, A, T. Reading from 5 prime to 3 prime. Cut is made between what, sir? Cut is made between T and C. Cut is made between T and C. Therefore, is this 8 types of restriction endonuclease enzymes, are they clear? Are they clear? Right. Now, these are some important statements. Listen carefully. Sir, on the board, I wrote 8 types of restriction endonuclease enzymes. Each and every restriction endonuclease enzyme is having its own recognition site, yes or no? Therefore, we are having more than 900 types of restriction enzymes. Each and every one is having its own recognition site or restriction site. Is this statement okay? Is it okay? Therefore, each type of restriction endonuclease having its own having its own recognition site, having its own recognition site. Is it okay? Right. Now listen here. Sir, these are 8 types of restriction endonuclease enzymes. Sir, these 8 types of restriction endonuclease enzymes, they are isolated from how many bacterial genera? See, they are isolated from 7 bacterial genera. Not 8, they are 7. Why, sir? Because PVU1, PVU2, these two are isolated from same bacterial genus. Isolated from same bacterial genus. Is this statement okay? Right? Therefore, what and what, sir? PVU1 and PVU2, right? Isolated from same bacterial genus. Isolated from same bacterial genus. Is it okay? Now see here, sir, this is 7, right? What? This 8 types of restriction endonuclease enzymes. They are isolated from how many bacterial genera? 7 bacterial genera, right? This here, this means I am speaking about this 8, okay? Therefore, this 8 types of what, sir? This 8 types of restriction endonucleases. 8 types of restriction endonucleases isolated from 7 bacterial genera. Isolated from 7 bacterial genera. Is this statement okay? And this statement is very, very important. Listen carefully. Sir, PVU2, out of this 8, except PVU2, remaining 7 are making staggered cut. Whereas PVU2 makes which cuts are even cut. You can check it. Yes or no? Therefore, it is cutting, right, both the strands at the same point. See here. Who? PVU2. Therefore, PVU2 makes which cuts are even cut, whereas the remaining 7 made which cuts are staggered cut. Is this statement okay? Right? Therefore, out of this 8 types of what? Restriction endonucleases. Right? Number 1, PVU2. What it is? PVU2 makes which cut? Even cut. Makes even cut. Number 2, remaining 7 makes which cuts are staggered cut. Remaining 7 makes which cut, sir? Staggered cut. Therefore, is this statements okay, clear, right? Therefore, this is concerned to which cuts, sir? Even cut and staggered cut, and these are some important restriction endonuclease enzymes. These are some important types of restriction endonuclease enzymes, right? Restriction enzymes, right? Now here we are going to discuss about some important restriction enzymes here, right? Eco R1. What it is? Eco R1. Sir, this Eco R1 isolated from E. coli bacteria, isolated from E. coli bacteria by strain RY13. Therefore, sir, this Eco R1 is isolated from, right, which strain of bacteria? That is E. coli. E. coli RY13 strain of bacteria, isolated from E. coli RY13 strain of bacteria. Is it okay? Right. Sir, what is the right recognition site or restriction site? I am speaking about Eco R1, right? Its restriction site is a GAATTC, reading from 5' to 3'. Yes or no? What is its restriction site? GAATTC, reading from 5' to 3', reading from 5' to 3'. Sir, where it cuts, sir? Sir, Eco R1 makes which cut? Makes a staggered cut. Makes a which cut, sir? Makes staggered cut. Makes staggered cut. Therefore, produces which ends, sir? As it is making staggered cut, produces which ends? Protruding ends or sticky ends or cohesive ends. Therefore, producing protruding ends or sticky ends or cohesive ends. Is this statement okay? Right. Sir, we are discussing right, Eco R1. Right, sir, it cuts between what and what, sir? G and A. Cuts between what, sir? G and A. Therefore, it is cutting in between what and what, sir? G and A. Cuts between G and A. Is this statement okay? Clear? Right. Now, I am writing a recognition site or restriction site of Eco R1. What it is? GAATTC, reading from 5' to 3', right? CTTAAG 3', 5'. Is it okay? Here what are present? Hydrogen bonds, right? Here which bond is present? Phosphoester bond. Okay, right? Now listen this statement. Sir, Eco R1, what it is? Eco R1. Sir, it is making a cut between what and what, sir? G and A. Making a cut between what and what? G and A. Yes or no? Right. Now see here carefully. Sir, this is a restriction site or recognition site of what, sir? Eco R1. This is the restriction or recognition site of Eco R1, right? How many base pairs it is having? 6 base pairs. Therefore, it's what? It's a restriction site with how many base pairs? 6 base pairs, BP base pairs. Sir, out of 6 base pairs, how many are GC pairs, sir? 2. How many are AT pairs, sir? 4. Therefore, out of 6 base pairs, 2 are GC pairs, whereas 4 are AT pairs. Is this statement okay? Right. Therefore, how many are GC pairs? 2 are GC pairs, whereas 4 are AT pairs. Is it okay? Right. Sir, in this recognition sequence, how many nucleotides are present, sir? In its recognition site or restriction site, how many nucleotides are present? 12 nucleotides are present. 6 pairs means 12, right? Therefore, it's a restriction site. It's a restriction site with 12 nucleotides. 12 nucleotides are present means how many nitrogen bases are present? 12 nitrogen bases are present. Sir, out of 12 nitrogen bases, how many are purines and how many are pyrimidines? 6 are what, sir? Purines. 6 are pyrimidines. 6 are purines and 6 are what, sir? Pyrimidines. 6 are purines and 6 are pyrimidines. Is it okay? Sir, purines means what and what? Adenine and guanine. Adenine and guanine. See here. Sir, 1, 2, 3, 4, 5, 6. Therefore, sir, 6 are what, sir? Purines. Purines means what and what? Adenine and guanine. How many G's are there? Guanines 2. How many A's are there? Adenines 4. Therefore, 2 guanines plus 4 adenines. Is it okay? Pyrimidines. Thymine, cytosine, T and C. See here. Sir, how many T's are there, sir? 1, 2, 3, 4. 4 T's are there. How many C's are there, sir? 2 C's are there. Therefore, 2 are cytosines and 4 are what, sir? Thymines. Is this statement okay? Clear? Right? Listen here. Sir, in its recognition site or restriction site, how many hydrogen bonds are present? You count the number of hydrogen bonds, they will be 14. How many hydrogen bonds? 14 hydrogen bonds are present. I am discussing about restriction site or recognition site of eco R1. Therefore, its restriction site. Its restriction site with how many hydrogen bonds? 14 hydrogen bonds. Sir, out of 14 hydrogen bonds, right, 6 hydrogen bonds are 3 hydrogen bonds, whereas right, 8 are 2 hydrogen bonds. Therefore, out of 14 hydrogen bonds, 6 are which bonds? 6 are 3 hydrogen bonds, whereas 8 are what? 2 hydrogen bonds. Is this statement okay? Right? Therefore, out of 14 hydrogen bonds, 6 are 3 hydrogen bonds, whereas 8 are 2 hydrogen bonds. Is it okay? Now listen this statement. Sir, eco R1, it is cutting in between what and what? G and in between G and A, what is present? Phosphoester bond. In between G and A, what is present? Phosphoester bond. Therefore, eco R1 is cutting here one phosphoester bond and here one phosphoester bond. Therefore, cuts phosphoester bond. In the book, it will be phosphodiester bond, right? But strictly speaking, it is cutting which bonds are? Cuts phosphoester bond. Cuts phosphoester bond. Be careful, a restriction enzyme always cuts which bonds are? Phosphoester bond. Therefore, restriction endonucleases. What? Restriction endonucleases. Restriction endonucleases cuts what? Phosphoester bonds. Restriction endonucleases, do they cut hydrogen bonds? No, they cut phosphoester bonds. Therefore, here how many phosphoester bonds are removed? You see, 2 phosphoester bonds are removed. Therefore, cuts 2 what? 2 phosphoester bonds. Cuts 2 phosphoester bonds. Now see this statement carefully. Sir, when it is going to cut here and here, is it okay? When it is going to cut here and here, then we are going to get fragments, right? Sir, this is one fragment, this is one fragment, whereas this is another fragment, yes or no? I am writing that fragments here, see. G, 3 hydrogen bonds C, T, T, A, this is one fragment. Is it okay? Right. Now go for another fragment. What it is? A, T, T, right, C. We got how many fragments here you see? We got 2 fragments. Sir, take this fragment, sir. Sir, here which bond is broken, sir? Phosphoester bond. Here which bond is broken? Phosphoester bond. Sir, when this phosphoester bond and this phosphoester bond are broken, automatically these 8 hydrogen bonds are removed. What are removed? 8 hydrogen bonds are removed. Phosphodiester bond, phosphoester bond. Is this statement okay? Sir, restriction endonuclease cuts phosphoester bonds, but not hydrogen bonds. When eco R1, a restriction endonuclease cuts this phosphoester bond and this phosphoester bond, automatically these 8 hydrogen bonds are removed. That's why you see here hydrogen bonds not there, here hydrogen bonds are not there. Is this statement okay? Clear, right? What? Eco R1, when eco R1 cuts, when eco R1 cuts a phosphoester bond, phosphoester bonds, hydrogen bonds, what? 8 hydrogen bonds, 8 hydrogen bonds are removed. Is it okay? How many hydrogen bonds are removed? 8 hydrogen bonds are removed automatically. But these hydrogen bonds are not removed by restriction endonucleases. Restriction endonucleases cuts phosphoester bonds. When it cuts phosphoester bonds, automatically 8 hydrogen bonds were removed. Sir, all 8 removed hydrogen bonds are 2 hydrogen bonds. Yes or no? All removed 8 hydrogen bonds are 2 hydrogen bonds. 2 hydrogen bonds present between what, sir? A, T. That 2 hydrogen bonds are present between what and what, sir? A and T. Present between what and what, sir? A and T. Now see here. Sir, this is one fragment, this is another fragment. Sir, this is which end, sir? Sticky end or cohesive end or protruding end? Yes or no? Therefore, in each fragment, the sticky end with how many unpaired nucleotides? 4 unpaired nucleotides. Sir, I will see this fragment, sir. You can check, no problem. Sir, this is another fragment, sir. In this fragment, right, the sticky end with how many unpaired nucleotides are? 4 unpaired nucleotides. Out of 4 unpaired nucleotides, right, 2 are A and 2 are, right, out of these 4 unpaired nucleotides, unpaired nucleotides, right, A, T, T, right. Therefore, out of these 4 unpaired nucleotides, right, 2 are A's and 2 are what, sir? T's. All these statements concern to what we are discussing, eco R1. Therefore, is this statement okay, right? Now listen here. Each sticky end with 4 unpaired nucleotides. Out of 4 unpaired nucleotides, 2 are A's and 2 are what, sir? T's. 2 are A's and 2 are T's. Is it okay? Right. Now, sir, I am speaking about recognition site or restriction site of what? Eco R1. Is it okay? Right. Now, listen this statement. Length What is the length of a restriction site of eco R1? Eco R1. What is the length of restriction site or recognition site of what, sir? Eco R1. How we will calculate it? First you tell me, in the restriction site or recognition site of eco R1, how many base pairs are present? How many base pairs? 6 base pairs. How many base pairs are present? 6 base pairs are present. Yes or no? Right. Sir, in first year we read that 20 nucleotides. How many base pairs? 10 base pairs. Therefore, what is the length for 10 base pairs, sir? If 10 base pairs are present, the length is 34 angstroms. In the restriction site or recognition site of eco R1, how many base pairs are present? 6 base pairs are present. 6 base pairs are present. Therefore, calculate the length. Therefore, how much it becomes? 6 into 34 by 10. You will get it as a 20.4 angstroms. Therefore, what is the length of a restriction site or recognition site of eco R1? 20.4 angstroms. If you convert it into nanometers, it will be 2.04 nanometers. 20.4 angstroms or 2.04 nanometers. This is the length of restriction site or recognition site of what, sir? Eco R1. Is it okay? Right? Thank you.